So today we're going to talk about a subject that's near and dear to my heart, the circadian clock. And I'm going to tell you about data integration strategies, pathways, and targets to modulate circadian clocks. So first I'm going to give you an outline or an introduction to clocks, and then I'm going to tell you about two integration strategies, one to find pathways that modify the clock, and a second to potentially find new clock components. And then I'm going to leave you with thoughts, challenges, and future directions. So the reason why we care about circadian clocks is that much of our physiology and behavior is clock regulated. And that includes things like our core body temperature. We have about a 1 degree centigrade rhythm in our core body temperature every day. And heart rate and blood pressure. Most heart attacks occur between 8 a.m. and 12 noon. Many of our hormones, including corticosteroids and melatonin, have a circadian rhythm. And then the functioning of many of our major organ systems, such as lungs and skeletal muscle, have a circadian rhythm. In the lower part of the panel, you can see actigraphy in a healthy human subject, where they have a watch, and every time they move, the watch registers activity, and the higher the black bar, the more activity they have. And so this is measurement of activity over time. And here you can see that this person's getting up at very close to the same time every day on Tuesday, Wednesday, Thursday, Friday, and Saturday, and so they have an apparently normal circadian oscillator. So as you all know, light is perceived by the eye. We have both visual and non-visual photoreceptors. So there's rods and cones which form images, and melanopsin, a non-image forming photoreceptor, that integrate light information and through the retinohypothalamic tract signal down to the suprachiasmatic nucleus of the hypothalamus, where the body's master clock is located. And inside each one of these SCN neurons is a cell-autonomous circadian clock that works in concert with other SCN neurons and to keep master time and integrate light information. So I talk about the SCN as being the master clock, and what it really is sort of an atomic clock. So it keeps the official time and integrates light information. And here you can see an individual SCN neuron that's been engineered with the luciferase vector and the incredibly robust rhythms in transcription that result over a period of 14 days. And these recordings are from David Welch at University of California at San Diego. And while the SCN contains the master clock, it also signals to cellular clocks throughout your body. So you actually have clocks in your fibroblasts, your liver cells, your lung cells, etc. And the SCN works to integrate the light information and relay this information to the rest of the clocks throughout your body in a somewhat unworked out process. So when I talk about the cell autonomous oscillator, this is basically our current understanding of what's called the core EBOX loop after about 20 years of research on both the fly and the mammalian system. So there are two transactivators, BMAL1 and clock, which bind to the repressor proteins, the period and the cryptochrome genes, activating their transcription. The purr and cry messages are then translated in the cytoplasm, where the purr and cry proteins complex with one another and with other factors such as the casein kinases, and the entire complexes eventually translocate back to the nucleus, where eventually they achieve high enough levels to interact with and pull the clock and BMAL complex off of DNA, shutting down the transcription of their own loci, the purr and cry loci, in addition to hundreds of others. Subsequently, the purr and cry proteins are themselves degraded, and clock and BMAL are then free to hop on their promoters and start another cycle. And this slide is actually courtesy of Joe Takahashi, who is a Howard Hughes investigator at the University of Texas at Southwestern. So our first question, true or false? The signal transduction pathway is cell autonomous, but behavior and physiology aren't. That's true. While signal transduction is cell autonomous, and you can study these processes in cells, physiology, such as, for example, cholesterol metabolism or control of locomotor activity behavior, are decidedly non-cell autonomous. So a few years ago now, in collaboration with Steve Kay, Andrew Liu, and Eric Zhang, we decided because the cell is cell autonomous, to try to figure out if we could work out new components of the clock doing genome-wide screening. And so together with engineers at Novartis, we designed and implemented a robotic system to screen in 3D4-well format for RNAi reagents capable of modifying the clock. And in this case, these reagents were designed against all protein-encoded genes, approximately 22,000 protein-encoded genes in humans, 4 siRNAs each. So what you're watching the robot do actually is take a 3D4-well plate and perform what's called a reverse transfection, where the nucleic acids are at the bottom of the plate, and cells, or first lipids and then cells are added to each well to complete the transfection process. And then the plate will go to what's called a hotel incubator, where it will remain for a period of time to acclimate. And then later, every 2 hours, a plate will be pulled from this hotel incubator and sent over to an imaging station where a reporter gene will be measured. And then actually the plate will be returned to the incubator. The process will start again every 2 hours. So all in all, it's 88,000 siRNAs, each measured every 2 hours for 4 days. Millions and millions of data points. So after we collect this data, we want to be able to analyze it. And so in this particular screen, we have 2 positive controls. The first positive control is for period length, which you can see up here. And what the control is actually Cry2. And when you add a Cry2 siRNA, the clock goes long period. So you can see here in U2OS cells, the period's approximately 25 hours. And when you inhibit it, it goes to about 29 hours or so when you inhibit Cry2. Conversely, when we're looking at amplitude, the amplitude is relatively constant throughout the screen, but when we add siRNAs against BMAL1, the only required component of the oscillator in vivo, we kill the clock. So here's our control data to the right, and on the left, you can see that most siRNAs have no effect on period length or the amplitude of the oscillator, but we have plenty that are causing the clock to go long or short period, or even some that are causing the clock to go high amplitude, and many things that are causing the clock to go low amplitude in our screen. So taking all this data into account, we wanted to ask a very simple question, is which pathways modify the clock? And so we did pathway-based gene integration of this gene expression data, but also our cell-based screening data that I just mentioned, and using SQL at the gene level and pathway information, we were able to determine which pathways are strongly intersecting with the clock. And we've verified these results by replication, performing these studies multiple times in different cell types, also using secondary screens and with tangential reagents such as small molecules. So here's perhaps the most interesting example from this work, which is the intersection of the insulin signaling pathway and the clock. So here's the insulin signaling pathway as defined by KEGG. There's about 40 components of the pathway, and shaded in colors are all the components that are either transcriptionally regulated by the clock, and those are in this sort of orange color or purple, and also all the components that, when you inhibit them, change clock function in our cellular model, which are in blue or purple. So we can see that approximately half of the components of the insulin pathway are under clock control, and approximately a quarter of the insulin pathway, when you inhibit those factors, it feeds back on the clock, mostly making the clock go long period, and that's shown over here. Because this pathway has been under such strong study by the pharmaceutical industry, there's a lot of small molecules designed against these components, and when we put these in to our same cellular assay, we're able to show changes in period length and changes in the phase of clock gene expression that match very tightly what we see with RNAi reagents. So even though you'll see many talks talking about the insulin pathway and maybe a few talks about the circadian clock in your sort of scientific endeavors, typically you'll see those talks without discussing that the two pathways are in fact highly integrated. So in a cell, in an organ, in an organism, these pathways are communicating information in real time. It's not just the insulin signaling pathway, the hedgehog pathway, the cell cycle, folate biosynthesis, and many other pathways are also integrated with the circadian clock and vice versa. Second question, yes or no? If there are 25,000 components in a pathway out of 25,000 genes, and 5 of 500 hits in your screen map to this pathway, is this pathway overrepresented in your screen? And the answer is yes. 0.1% of the genome is in the pathway, and 1% of your screen hits are in the same pathway. So it is overrepresented by tenfold. So this also complicates having this amount of data and this potentially rich set of new clock modulators complicates the biology. So before the RNAi screen, our picture of the clock was actually relatively straightforward. This relatively straightforward core E-box loop and ROR box loop, which I did not mention. And after the screen, we have a much more complicated and set of components that may be components of the clock or simple modulators. And so a very straightforward question is, which of these are clock components? And so here, to get at that, we're going to use network or mechanism-based integration, integrating known interactions, both protein-protein interactions and transcriptional interactions from the literature, and also integrate new protein-protein interactions from Prolexis and then the NCBI to define potential new edges, and integrate that with the cell-based screening data to define functional consequences, functional new edges and nodes in the clock network. So here's our version of that integrated network. The components here in the center, which are indicated in blue, are all known clock components, and the rest of the molecules are hits in our screen, which are depicted in green or in red or in purple for high amplitude hits. And protein-protein interactions are drawn in black. We used bridging interactions. So we allowed for one component between the circadian clock component, for example, here, Cry1, and this hit from the screen, IRF4, and that's TRAF2. So there's other ways you can do this type of integration. So here's a probabilistic or machine learning approach. So we know a lot about clock factors from studying them for a long time. Number one, they cycle at the mRNA level. Every single mammalian clock member cycles. It may not be everywhere, but it'll cycle somewhere. They tend to have phenotypes in cellular models. They tend to physically interact with one another in protein complexes. They tend to have Drosophila orthologs, and they tend to be expressed nearly everywhere in your body because there are clocks nearly everywhere in your body. And we're going to try to codify these rules. No rule is absolute, and each data set should have predictive value. And to do that, we're going to use something called naive Bayesian integration. These methods were first sort of applied to biology by Olga Troyanskaya, working with Russ Altman and David Botstein at Stanford. And it's basically identifying a metric that encapsulates each of these criteria, computing a Bayes vector, which is basically the amount of evidence that each result provides, indicating that a particular gene is a core clock component. And then we're going to consider all these Bayes factors independently and come up with a sum. So here's a distribution of p-values given in red or in blue here for all genes in the genome or core clock genes. So these are plotted in the same axis, which is log p. So we go from minus 120, extremely highly cycling, to 0, not cycling at all. And you can see that most genes in the genome don't cycle, but there's this tail out here of genes that do cycle. And in fact, if you plot just clock genes on the same graph, you can see that clock genes are way overrepresented in the cycling category. So this actually gets a relatively high Bayes score, and we're going to simply do the same thing with other data sets that encapsulate what we know to be true about clock factors. So here's the RNAi screen that I mentioned earlier. Here's the distribution of all genes in the genome for having an impact on our cell-based screen, and here's the distribution of core clock genes. So there's information that's contained in the RNAi screen that's indicative of core clock factors. There's information in genetic interactions. There's information in looking at tissue ubiquity. I told you that clock genes are expressed nearly everywhere. Here's a codification of that, looking at tissue ubiquity. And at the end of the day, it boils down to a table. Gratifyingly, we found many of the core clock factors. It's sort of circular logic because we use these to actually train our algorithm, but we also find a lot of genes that are not known to be core clock factors down here, and in particular I'm going to highlight this GM129 or C1 or 51 as it's known in humans. So why not integrate every experiment you can? You could overfit, i.e. unimportant details are rewarded. For example, clock, cry1, and cry2, all bona fide clock components all start with the letter C, but starting with the letter C has nothing to do with being a clock component. B, it would take a lot of people, time, and money to integrate every experiment you could. C, just A and B, or D, you should integrate everything. It's a good idea. The answer, of course, is A and B. Although clock, cry1, and cry2 all start with the letter C, that is purely a coincidence. It would take a lot of people, time, and money to integrate all possible data sets, and it may not be worth it. In fact, you can throw in poor data sets, and at best, they will not help it. And at worst, they could actually hurt your integration experiment. Of course, all these integration methods are basically hypothesis generators. So it's really important that you have a follow-up strategy to experimentally validate new hypotheses. And here our experimental validation strategy is going to be to evaluate protein-protein interactions with known clock factors, to test these new factors in cell-based assays of clock function, to test them in other circadian cellular models, and also to do either lentiviral experiments in SCN tissue slices or behavioral analysis in knockout mice, which is really the gold standard for the field. So here's a mammalian 2 hybrid experiment looking at protein-protein interactions. On this side, we have Gal4 DNA binding domain vectors. On this side, we have VP16 activation domain vectors. And when the two proteins interact, you have a red signal or a transcriptional induction. And when we tested GM129, or a gene we're now calling chrono, we found that it strongly interacted with PER2 and BMAL1. And here it is interacting with PER2 and BMAL1 as a VP16 fusion. Over here to the right is basically a graphical representation of the chart on the left, where you show Gal4 chrono interacting with VP16 tagged PER2 or BMAL1. And down in the lower left-hand corner, you see the interaction of Gal4 tagged chrono with with BMAL in conjunction with VP16 tagged clock over here. So this is sort of the native form of BMAL. And in the lower right-hand side, you see IP mass, basically IP Western blots, where we have tagged versions of the activators clock and BMAL and chrono, and you can see that there's a strong interaction between chrono and BMAL1 and BMAL1 and clock over here on the right-hand side. Subsequently, we've learned that chrono is interacting with BMAL and clock independently of the interaction surfaces in which the cry repressors are interacting with them. So here you can see that here's a version we've evolved of clock and BMAL1 that have point mutations that render them relatively insensitive to cry1, and Chrono is still capable of repressing their functional activity as strongly as it does to the wild type form. So here's Cry1, which is a strong repressor of clock and BMAL. Here is Chrono, which is a strong repressor, less strong than Cry1, but still a strong repressor of clock and BMAL. And in the Cry insensitive clock and BMAL versions, these point mutations 612 to E or H360 to Y of clock, Chrono still works practically the same way. We've also shown that inhibition of Chrono influences circadian rhythms and reporter gene expression in a cellular model. This is NIH3T3 cells, where we show that Chrono inhibition is capable of changing clock function in cellular models as well. So question 4, validation is important because A, unvalidated hypotheses are cheap. B, validation experiments can point you at potential mechanisms of action. C, new data could be used iteratively to refine your integration strategy. D, all of the above. And E, B, and C. And the answer is all of the above. Unvalidated hypotheses are cheap and you could conceivably come up with them in many different ways, and they're actually not of much value unless someone, either you or someone else, is willing to actually test and validate them. The experiments themselves can actually tell you about the potential mechanisms of their action. For example, the protein interaction studies, if you figure out that they interact with one particular protein, maybe they're modifying their post-translational status, their cell location, etc. And new data sets that you could get in these validation experiments could in turn be used iteratively to refine your integration strategy. So all of these answers are correct. So final thoughts. I think you really need to know what you're looking for and have good examples. Nothing beats good data analyzed properly. So if you have poor data and it's analyzed improperly, you're really not going to have a lot of separation between your old components and the new ones that potentially exist. We look to encapsulate expert intuition. So try to find data sets and metrics that can take what the human brain is great at, which is insight, and really algorithmically encode them. It's super important to have a well-developed validation plan. If you don't validate these hypotheses, then I would argue that they're, if no one does, they're essentially worthless. And if you don't, and you feel strongly about them, it's sort of a lost opportunity. And I also think you need to temper your expectations when thinking about these data integration strategies. The questions I'm asking, is this gene a potential component of a system, is a relatively simple question. If I were to ask a different question, what's the best step in a pathway that will make a good therapeutic target, that's a much more difficult and nuanced question. And maybe these methods, because we don't know enough about those processes, maybe these methods really won't work at this particular juncture. So final slide, I'd like to thank the people that did the work. Ron and Afi did all of the Bayesian integration that I mentioned earlier. The screen was a collaboration with Andrew Liu, Steve Kay, and Eric Zhang. And this work was funded by the National Institutes of Health and by DARPA.